or maybe we can have Devina introduce Katie.
Yeah, I think so. I think it's
Yeah.
much
Okay.
better. Yeah.
Yeah, sure. I'd be happy to.
Yeah.
Okay, so I'll introduce Katie Diop. So Katie has been in my research lab for about a year and a half. So she's actually going to be starting as a PhD student in the fall with us. We're really excited to have her. And Katie's research focuses on looking at reprogramming strategies, especially developing in vitro models to look at the effects of reprogramming on human disc cells and trying to reverse that degenerative phenotype. So please take it away, Katie.
Thank you. So today my presentation will be on non-viral reprogramming of the human IVD cells using developmental factors. So why should we care about the spine and the disc in general? 40% of low back pain is caused by complications of the disc, and back pain is the leading cause of disability worldwide, and it will affect 80% of the population. Many of the current therapies that we have involve invasive surgeries or therapies that are just not as effective. So it's important that we come up with minimally invasive treatment options that improve both structure and symptoms of back pain. The human intervertebral disc can be characterized into three distinct tissue types. There's the nucleus pulposus, which is the metabolic core of the disc. This is comprised mainly of proteoglycans, which gives it a gelatinous texture. Around the NP, there's the annulus fibrosus. These is concentric collagen rings that surround the NP and give the disc structural support. Not pictured here, there's the cartilage end plate, which is a layer of cartilage that can be found above and below the disc that allow nutrients to diffuse. Disc degeneration can happen either with time or with injury, and it's defined as an aberrant cell-mediated response to structural failure. With degeneration, you'll see structural loss of distinct regions in the disc, and with the highest degree of loss corresponding to the highest grade on the Thompson and the Furman scale. Because the disc is avascular, it has a hard time repairing itself. The degenerate IVD is a very hostile environment, so it makes it quite difficult to repair. Some of the considerations we have to account for are the very few cells that are in this landscape, the limited hydration. Because of complications with the end plate, there's limited nutrition, and there's also excessive inflammation. So given these challenges, how can we repair the degenerate IVD? One common approach to cellular reprogramming is the use of induced pluripotent stem cells by using Yamanaka factors. However, we're unsure if these stem cells can survive in the hostile environment of the degenerate IVD. This is kind of where our idea of cellular reprogramming comes into play. One of our collaborators, Dr. Daniel Gallego Perez, was able to demonstrate using techniques like tissue nanotransfection, the ability to reprogram an adult somatic cell into a newly differentiated cell type. They named this term direct reprogramming. So we wanted to try and apply this concept to the IVD cell and have this revert back to a healthy IVD cell displaying like the normal functions and structures in a healthy state. So after this, we had to decide which developmental factors to use. We wanted to use factors that are specific to the tissues as they all have distinct properties. So for the NP region, we decided on brachyury and FOXF1 because of their general role in development as well as being healthy markers for NP cells. When approaching the AF tissue, we wanted to use Mohawk and Scaraxis for a similar reason. They're both involved in development, maintenance, repair, as well as regeneration of AF tissue. So these are great candidates for IVD cells. After deciding on which factors we wanted to use, we then had to decide on a delivery system. A common delivery method is viral transfection. However, viral transfection can have issues with mutagenesis or an unwarranted immune response. So we wanted to use a non-viral delivery. There are a number of non-viral delivery methods, but we want to focus on bulk electroporation, namely because of its feasibility and high efficiency in vitro, as well as engineered extracellular vesicles. EVs are great because they deliver genetic cargo in vivo. They're non-immunogenic because they're host-derived and self-generated in vivo. And it's been shown that mesenchymal stem cells, as well as notochordal cell-derived EVs, have a regenerative effect on IVD cells. So knowing this, we want to know if we could use this delivery system to deliver our transcription factors of choice. We were able to demonstrate in previous work that we can reprogram IVD cells back to a healthy phenotype using brachyury to target the NP specifically. We were also able to demonstrate that FOXF1 reprograms diseased human NP cells similarly to brachyury, and that we can use FOXF1 loaded EVs to target IVD cells in vitro and in vivo. Currently, we're interested in the synergistic effects of FOXF1 and brachyury, so we want to know if these two plasmids, when used to treat IVD cells together, have a greater effect of reprogramming. So we'll generate these plasmids and then, we'll generate plasmids and then transfect human NP cells to generate EVs. We'll then treat a 3D hydrogel culture with the EVs or a control and then assess variables like gene expression, collagen, protein, and viability at both 2 and 8 weeks. This is an ongoing study, but so far we've been able to show that brachyury and FOXF1 are both upregulated in the synergistic groups at 2 and 8 weeks, and that there's a significantly higher amount of collagen at the 8-week time point in our synergy group. So far, our trend is that brachyury and FOXF1 loaded EVs exhibit better reprogramming abilities compared to the factors alone. In addition to looking at the synergistic effects of FOXF1 and brachyury, we're also interested in increasing the efficiency of EV uptake. So in this case, we're trying to determine if attaching a ligand to the outside of the EVs will allow for preferential uptake of EVs to allow for cells tissue specificity. In this case, we've been able to show that NP cells show preferential uptake of CD11A functionalized EVs compared to the non-functionalized group, and that AF cells show preferential uptake of netrin functionalized EVs compared to their non-functionalized counterparts. We're also interested in reprogramming the AF tissue, and in previous work, we've been able to demonstrate that, Mohawk or scleraxis can reprogram a moderately and severely degenerate AF cell towards a healthy phenotype. We're now interested in the synergistic effects of Mohawk and scleraxis, similar to our NP synergy study. In this case, we'll use the EVs to treat a cell culture that is seated on a 24 nanofiber well plate. These fibers are either organized or disorganized because we're also interested in the microenvironment and its impact on the cell's ability to be reprogrammed. So in this case, we're analyzing similar variables where we're doing gene expression, collagen, protein expression, as well as cellular morphology. This is also an ongoing study, but so far we're able to see clear morphological differences across the fibers, and that the cells carry a more like standard fibroblast-like shape on the organized fibers compared to the branch shape on the disorganized fibers. We're performing our functional analysis of our RNA-seq data currently, but some of the comparisons that we're interested in are in the disorganized and organized groups across the control groups to kind of parse out the effects of substrate. We're interested in synergy versus control on the disorganized as well as the organized fibers. And then we're also interested in the synergy of groups on the disorganized compared to the organized fibers. So far, we have a few trends that we've been able to parse out. So when comparing our control groups across substrate, we see a decrease in the immune and inflammatory response, as well as an increase in matrix remodeling. When we compare our synergy on the organized fiber compared to the control group on the organized fiber, we see that a lot of the notable pathways map to pain and neuronal sensitization pathways. When comparing our synergy on the disorganized versus the control disorganized, we see a decrease in pathways that are involved in osteoarthritis. And when we compare the synergy disorganized to the synergy organized, we see a similar trend to the control group, where there's a decrease in the immune and inflammatory response and an increase in the matrix remodeling. So far, our work shows that EVs have great potential for non-viral gene delivery, and our previous work shows that our ability to reprogram diseased IVD cells back to a healthy phenotype using this delivery system. In the future, we'd like to test the translational effects of reprogramming by using these therapies in an in vivo model, which Dr. Purmessur will talk more about. As far as NP synergy, we'd like to continue examining the effects of sex and age on gene and protein expression, And for our AF synergy project, we like to continue examining collagen expression, cell morphology, as well as finishing our functional analysis of the RNA-seq data. So our acknowledgements are to NIH for funding our projects, as well as OSU. And then I'd also like to acknowledge the Purmessur Lab for assisting with this project, as well as Nina Tang for helping design some of these projects. And of course, our lab family.
Thank you, Katie. It was a great presentation.
Thank you.
So does anyone has any questions for Katie?
Hi. Yes, I have a
Yes.
question. Thank you so much for a great presentation. I'm just wondering, how can we differentiate the functional and non-functional EVs accurately, actually, in the molecular level?
Like the functionalization. So in this case, we just looked at the degree of uptake. So we had they did imaging to see how many cells were being local, or how many EVs were localizing to cells compared to the functionalized EVs.
So it can be achieved through the functional scan. Am I right? It's like,
I'm not sure which kind of imaging they did to determine this, but I think it was was it TEM, Dr. Purmessur, or SEM?
So in terms of the uptake, it was just fluorescent imaging that they did. So they labeled the EVs and then they co-cultured them with NP and AF cells. And then they wanted to look at the ones that, after labeling, look at the difference in terms of uptake.
yeah, thank you so much. Thanks.
Sure.
Thank you.
Katie, that's a wonderful
Yes.
presentation.
Yes, Dr. Foxon, please go
Sorry.
ahead.
Yeah, wonderful presentation, Katie. So about the EVs, how are they prepared? Was it just centrifugation and then you isolate certain EVs, or?
Yeah, so they bulk electroporate healthy NP or healthy fibroblast cells with the plasmid of interest, and then these EVs will be released into media containing the plasmids that we want, and then they're isolated and collected and quantified, and then they spin them down using nanocentrifugation, and then we use them to treat our cell cultures.
Okay, thanks.
Thank you for your question. So
I have one more actually. Yeah, sorry. Yeah, Katie, yeah, it was a very, very nice presentation and amazing data. And I really like the approach of using EVs for delivery. So just a couple of questions. One of them is, for future studies, when you move on in the delivery of the EVs to the NP or AF cells, do you envision a problem when you're injecting, let's say, in the native tissue, or in terms of uptake, that may affect or maybe decrease the uptake? And how would you kind of address the amount of EVs you'd use for that?
So the number of EVs was determined based on pilot data. So they figured out a number that would be actually reflected in reprogramming, and also the native tissue part of the second part of the study will be functionalizing the EV so that they have increased uptake of whatever tissue type is appropriate.
And that's fantastic. And the second question, for the challenging the approach of how FOXF1, or if you introduce brachyury in EVs in your delivery, did you test whether, let's say, an inflammatory milieu or some degenerative milieu may affect, or how does the let's say delivery of FOXF1, how does that work when you change the environment a little bit to see how, and challenge the cells and see how they respond in that different environment?
Like in vitro, if we're just assessing inflammation.
Yeah, for instance, yeah.
So we'll do for NP Synergy, for example, will do PCR and assess like IL-6 or a number of inflammatory markers, and we'll do like a protein expression assay to determine the amount of protein that's being released. So yeah, we'll determine the effect of reprogramming to see if it's like decreasing an inflammatory response as well.
And I think, Michael, you're curious of whether we've tested it in actually an inflammatory like culture, right? Whether we have immune cells or so. We haven't done that specifically. So with the AF Synergy, we're starting to look at substrate, right? So it's the idea of using the disorganized versus organized as mimicking the kind of, at least with matrix environment, how it changes with the disease. But the work I'm going to show you in a little bit, we were introducing the EVs in a mouse model of back pain. So it's already got it's at the same time as inducing an injury, right? So we're providing that injurious environment. We haven't tested it in vitro, but that's a great point. It'd be really interesting to see if it can actively suppress some of those like potential immune stimuli or inflammatory stimuli. But I think that's a really good point for sure.
Thanks.
I have Muskan, I have a question.
Yes, please go ahead.
Thank you, Kadi, for this great presentation. At the end, you said that for the future work, this will be great potential for non-viral gene delivery. And for in your previous work, you show your ability to reprogram diseased intravertebral disc using a delivery system. Which kind of delivery system you are using in your previous work, and for the future, what you plan? Which kind?
So most of our previous work has been done using EVs. We've also done some studies that directly use bulk electroporation, but this is only feasible in vitro. In the future, we'll use EVs. We'll continue to use EVs, and hopefully we'll have functionalized EVs so that they have more specificity.
Thank you.
Thank you for your question. So do we have any more questions for Katie? Okay, if not, I would like to thank Katie for thank you so much for presenting at our seminar and taking your time. And yeah, and I would like to introduce Dr. Foxen Molly, our departmental head.
So thank you very much, Muskan, and thank you, Katie. So introducing our keynote speaker today is truly a privilege. Professor Devina Purmessur Water is a tenured associate professor in the Department of Biomedical Engineering in the College of Engineering at Ohio State University. With an impressive research training and a diverse scientific background in musculoskeletal pathology, she has developed a unique multidisciplinary program, which is funded both by the NIH and NSF. So her work stands at the crossroads of engineering, medicine, and veterinary medicine, showcasing her ability to build these bridges through clinical and research collaborations. Her pioneering research is driven by a commitment to address the unmet clinical need for non-addictive biologics to treat low back pain and regenerate the intervertebral disc with a solid foundation in intervertebral disc pathophysiology, developmental biology, mechanotransduction, and preclinical models of low back pain acquired from her graduate academic training at the University of Manchester. You can tell from the accent. Industry training at Pfizer, and further training in the spine bioengineering labs at the University of Vermont and Icahn School of Medicine at Mount Sinai in New York. So she's at the forefront of developing transformative biological strategies for back pain treatment. Now, beyond her research, she's deeply committed to teaching, mentoring, and fostering an environment in independent thinking, problem solving, and creativity. Her NSF career award-funded outreach program called Skeleton School. It targets preschool and kindergarten aged children, focusing on STEM and the workings of our musculoskeletal systems, reflecting her dedication to inspiring the next generation. Our service extends to supporting a diverse and inclusive learning environment at both the university and scientific community levels. She has played significant roles in the Louis Stokes Alliances for Minority Participation Program, NSF, RU, and the Women in Engineering Graduate Council, and has contributed to the advancement of professional development, education, and networking opportunities through her roles in the Spine Research Section, Community, and the Orthopaedic Research Society. So please join me in welcoming a visionary scientist, an inspiring mentor, and a dedicated community leader whose work not only advances our understanding of the spine as a whole organ system, but also paves the way for addressing complex real-world problems at the intersection of engineering and medicine. So thank you very much, Devina, for agreeing to come and talk to us.
Well, thank you so much. That was such a kind and generous introduction. Thank you. And it's such a pleasure to have the opportunity to talk to you about some of the things we're doing today. So thank you again. And so the focus of today, I'm going to be talking about some of the models that we use and the relevance of these models for disc research. So I won't spend too much time here, as Faxon gave a lovely introduction, but my lab, our lab community, we really do focus on diversity, equity, inclusion, really providing a nurturing environment for our students and staff. We're really interested in paradigm shifting ideas related to spinal disorders, so engineering treatments that target degeneration of the disc joint, looking at tissue crosstalk, inflammation and pain, and using different drugs and gene therapy systems. We're really fortunate in our lab to be able to work with translational models. So we span from using human cells and tissues, bioreactor systems, through in vivo small animal models, and then large animal models of pain. We're also very fortunate to be in an environment where we can look at and collaborate with orthopedic spine surgeons, sports medicine, and veterinary neurosurgeons. So we're really at the cusp of translation. And a big interest of our group is trying to get involved in outreach in our community. And so through various outreach events, we've been able to develop programs that really bring STEM and musculoskeletal STEM to our younger pre-K and kindergarten communities. So, but it takes a village. So I really, kudos to the lab. They really do the hard work. So I'm really appreciative of them. So yeah, so we're interested in looking at the disc as a whole joint system. So Katie gave a really nice introduction in terms of the disc joint, its structure, and we're focused at looking at disease mechanisms of the joint. So one area of interest for us is this region called the cartilage end plate, looking at its interactions with the disc tissue and subchondral bone, looking at it, kind of phenotyping it, and then looking at its role in mechanobiology. And then also in terms of crosstalk, looking at infiltration or extraspinal cells such as immune cells and how they can infiltrate in disease and play a role in inflammation and pain. And then we're also, a major focus is engineering therapeutics. We have a big interest in gene therapy, including delivery systems such as extracellular vesicles, and also using relevant models to look at and test our therapeutic strategy, which will be the focus of my talk today. So developing models for therapeutics. So what we're going to cover are small animal models used in disc research. So the mouse model, focusing on injury models. We're then going to talk about preclinical models, and a major focus for us is using chondrodystrophic dogs. Last but not least, I'll talk a little bit about some of the human tissue, human cell research that we're doing in the lab. And as I mentioned, it really does take a village, and it's really the hard work of the students and staff in the lab that drive a lot of these projects. So thanks to them. So in terms of mouse models for intervertebral disc degeneration and back pain, genetic and aging mouse models have been utilized. So Laura Stone's group have utilized the spark null knockout and being able to investigate different mechanisms and effects. In terms of aging, Chichida here has done some lovely work in terms of examining the age-related effects on disc degeneration and back pain. And then combining the two, both aging and looking at genetics, Mac Risbud's group has looked at inbred strains and how they show phenotypic diversity with aging. And then we're able to test different drugs, so senolytic drugs on these age-dependent kind of mouse models. So aging and genetic models are pretty common and widely utilized. And then in terms of injury mouse models, so it was early work by Dawn Elliott's group looking at the effect of needle puncture and how you can use needle puncture to really change the structure and function of the disc and induce disc degeneration. And this needle puncture injury model has also been used to look at potential mechanisms. And so there's nice work by Svenja Ilien-Junger's group looking at kind of the role of FLIP using an injury model to explore this. Injury models have been great in terms of characterizing the mechanisms of pain. And so Laura Stone's group has looked at the effects, looking downstream at innervation and macrophage infiltration in injury models. And then this has also been characterized by Im Sampen's group in Rush in terms of developing relevant models of actual pain. And our group did a kind of literature review paper, kind of trying to summarize some of the models that are currently utilized in disc research. And I think there's so many advantages, right? Clearly mice are not humans, but I think I really want to highlight that they're a great tool to investigate mechanistic questions, right, and understand disease. So as you can see here in the center, there's so many similarities at the cell molecular level, structure, function, and pain in both degeneration, which is where the X's are marked, versus health, right? So many similarities, and there are differences, but I think the advantages definitely outweigh the limitations of the mouse model. And I think what makes the mouse model really attractive is the fact we can assess multiple parameters just using the mouse, right? We're able to assess pain behaviors, both at the kind of macro scale and then looking at the dorsal root ganglion, looking at the neurons, phenotyping them. And then at the same time, we can look at structure and function of the disc. How is it changing? Using different imaging techniques, using mechanical testing, looking at protein level. And then when we get to the cell and molecular, we can start phenotyping the cells and kind of seeing what's going on at that end. So I think there's definitely many advantages of using the mouse model for disc research. So why use the mouse model? So just highlighting some of the advantages of using an injury model. What are some of the advantages? By inducing an injury, it accelerates degeneration. So this is a good rapid way for assessing degeneration. We focus on the lumbar discs because they're most physiologically relevant. The mouse models, especially injury model, is accessible by researchers all over. So that's very important. And then repeatability. So finding a model where you can do a repeatable injury that can be created in multiple labs is essential. And one of the nice things about using the mouse model is you've got so many genetic variants and so many different types of genetic knockouts. So using this model in terms of exploring questions is very attractive. However, that being said, there are limitations and challenges with any model we use, and I think we need to acknowledge that, right? So injury would be seen as more of an aggressive intervention. It doesn't necessarily recapitulate all aspects of human disc degeneration. We use an anterior approach, so we go in from the kind of the front of the mouse to access the lumbar disc. So this can be considered an invasive surgery. And also one thing to bear in mind, you induce an injury, and then when you go and intervene again with your treatment, this involves a second surgery, right? So you've got another injection into the disc, and that can be problematic if you've already got scar tissue. So some challenges remain, but definitely some advantages. And so in terms of our lab and how we've used the injury model, so we expose, this is our injury back pain model. We basically expose the lumbar discs, L4, L5, L5S1, L6S1, in both female and male mice. We look at mice who are about 15 weeks of age. We puncture the discs using a 30 gauge needle, and typically we'll inject sterile, about 2 microliters of sterile saline as we're doing the injury. We've seen similarities in structure and function of what we'd expect to see in a degenerate disc. So on MRI, we see a decrease in signal intensity. So this is a measure of tissue hydration. So we're seeing a decrease in tissue hydration, water loss, which is indicative of a degenerate disc. And then looking at pain behaviors, we separate out males and females. But what we can see with the females is that we see a decrease in grip strength in our injury group, which is suggestive of more pain. We also see a decrease in the distance that the female mice travel, also associated with pain in our injury group. The males, we see different parameters where we're seeing increased sensitivity. So here, looking at thermal hyperalgesia, so their sensitivity to cold is enhanced with injury, and then we perform the tail suspension, and this is associated. So if mice are more mobile, then they're in more pain, and so here we see an increase in the injury group of time mobile for the mice, suggesting they're in pain. We then isolate the dorsal root ganglion, which is the source, the neurons, which is the source of pain. And then we've done transcriptome analysis of both males and females separately. But some interesting findings we see in the females is we see an upregulation of kind of pain peptides, neuropeptides, neurotransmitters, also associated with neural regeneration, outgrowth, and injury. And in the males, we see more of a protective response. And that this is our initial kind of IPA analysis that we've done to probe that. But yeah, just wanted to highlight some of the overall model that we're using. And then in the next few slides, I'm going to show you how we're using it for therapy. So as Katie nicely summarized with some of her in vitro work, we're really interested in using non-viral gene delivery as a mechanism of, or a way of treating back pain, right? So now we have our model that we've identified, this injury model. We wanted to explore the effects of delivering developmental transcription factors. So in this case, it's FOXF1 to the injured disc in vivo. So this is the work with my collaborators and former PhD student, Nina Tang. So I just want to highlight them and their contribution to this work. So we generated extracellular vesicles from primary mouse embryonic fibroblasts, and we loaded them either with FOXF1 plasmids or a sham plasmid called CPMV6. We then isolated the EVs, and then we did a different kind of quantification looking at quantity, size, and the cargo. So in terms of characterization, we saw there was no difference between our control sham EVs and our FOXF1 EVs in terms of particle count. In terms of particle size, again, no difference. And with when we looked at our donor cells with FOXF1, we could see there was significantly more FOXF1 expression, which was to be expected in our donor cells with FOXF1 versus our sham. And then last but not least, in the actual EVs, with FOXF1, we could see significantly greater expression versus our sham control. So everything looked great in terms of characterization. We then, using our injury model that I just described to you, we injected or had the following treatment groups. So we had a FOXF1 EV group, a sham EV group, just an injury, which was a saline injection, and then we had a non-injury control. And so in terms of dependent variables, at 12 weeks, we did imaging, we assessed for protein, we did histology, we did mechanical testing, and then we also looked at pain behaviors. So in terms of looking at kind of initial structure function, we did MRI to look at signal intensity as a measure of tissue hydration. The control is nice and white, bright, high in signal intensity, and then injury, we see a decrease, but then restoration with our FOXF1 group. We quantified the signal intensity, and what we saw was an increase in signal intensity with the FOXF1 group compared to injury. So demonstrating that we're getting some restoration of this tissue hydration. We then performed micro-CT to look at disc height, and these are from L5 to L6. You can see where we injected or treated the disc, depending on the control, injury, sham, and FOXF1 group. And when we quantified disc height, we could see a restoration. Here, there's our FOXF1 group, restoration of disc height, similar to control and significantly different from injury with our FOXF1 treatment. So this was really exciting for us too. And it goes nicely with our tissue hydration data. So then looking at structure function, we want to look at the extracellular matrix, right? So looking at proteoglycan and collagen. So we performed histology on discs from our different groups. So here we have control. You can see the nice hydrated disc. This is alcine blue, picocereus red, where the collagen is red and the proteoglycan is blue. So control looks great. Injury, you can start to see collapse and disorganization, a thinning of this nucleus pulposus proteoglycan band, but we see restoration with our FOXF1 group similar to the control. We then quantified the amount of proteoglycan using the DMMB assay, and we see an increase in proteoglycan with FOXF1. And interestingly, we saw here an increase as well with our sham EB group. So it seems like the sham EBs also had some restorative effects. We then, in terms of structure function, we wanted to look at kind of the mechanical properties, whether we get restoration of, the mechanical properties of the disc tissue. So with this, we recruited another graduate student, Karl Kaczynski, and then Dr. Ben Walter to help us design and validate mechanical testing of mouse motion segments. And just so you're aware, these are very tiny, and this was really challenging to do. It was not easy. And so we have it hooked up to an Instron, and you can see there the mouse motion segment. But it was pretty exciting. It took a while to actually develop this device and get it working. But eventually we got the metrics that we needed. So this is just an example of the stiffness threshold method. And then we didn't see any effects when we looked at neutral zone range of motion, but when we looked at the threshold neutral zone stiffness, we did see an increase in stiffness with our FOXF1 group, which was really nice with the other parameters in terms of structure function that we looked at, that we're getting some restoration of the tissue repair here. Okay, and so we were also curious looking at FOXF1 expression, right? Where this is what's in our EBs. We were curious in terms of, looking at it staining in the disc tissue. Interestingly, what we saw was an increase in FOXF1 expression in both our injury and FOXF1 groups. We weren't sure whether in injury it's associated more with a fibrous, sort of frustrated healing. And then it was very focal for us in the FOXF1 group in terms of the cells that were surrounding the NP, including the end plate. And then now moving on to some of the pain and behavior outputs. So we looked at grip time, which is a measure of grip strength. And here you can just see kind of what the setup looks like. We separated out the males and the females. The control and injury groups are in the colors, so red for the female, blue for the male, and then our treatment groups are in black, so the dashed lines are our sham EVs, and then the solid line, black line, is the FOXF1. But what you can see is that with FOXF1 and the sham, we see an increase in strength, particularly in the female group compared to our controls, and not less so in the males. We then performed another assay. It's called the tail suspension assay, and this allows us to kind of look at effects on axial low back pain. So basically, if the mice are more mobile, that means they're in more pain, so they'll move more. Interestingly, over time, we saw a general decrease in pain over time for both males and females. However, interestingly, we saw a decrease in axial discomfort, and it was particularly evident for the male mice. We then looked at thermal hyperalgesia. So we do a cold plate or look at a cold plate stimulus and the effect on the mice. Again, separating out males and females. And here what we see is some real sex dependent effects. So we see decreases in sensitivity in the male mice with FOXF1, but we see the complete opposite with the females, which just emphasizes real importance to look at when you're looking at pain behaviors, to look and separate groups out based on sex. And then last but not least, we looked at activity. So just looking at the distance traveled, the mice are in this arena. And so we video them and see how active they are, and we can get certain metrics. So in terms of distance traveled, we definitely see, again, sex differences. There's a trend in distance traveled with the females versus the males. So in terms of take-home messages from our in vivo studies, we see that FOXF1 delivery by these engineered extracellular vesicles can restore structure function. And we looked at matrix, NP hydration, mechanical stability, and also decreases in pain. We also saw an interesting effect with our EVs, the sham EVs. So these were just empty plasmid, but the literature does point towards innate EVs, naive EVs having some restorative effects. So we put it down to that likely. And then these sex differences are really interesting and just really highlight the need for us to kind of look at males and females differently when we're analyzing the data and consider this in regenerative therapies. And then what we're doing right now. And what we're doing right now is we're looking at, so this model that we use, the injury model, so we're basically doing the treatment at the same time as injury, right? So that's more of preventative, but we really want to mimic the clinical situation. So we're currently developing a model right now in which we can intervene and it's more clinically relevant, right? And so we're also hoping to isolate the DRGs from these mice. So in addition to the pain behaviors, kind of assess what's going on at the neuronal level as well. And then in terms of EVs, we're doing a lot of characterization, improving purity, looking at cell uptake, and then biodistribution as well. Okay, so now some additional questions, and Katie kind of alluded to this. So we're reprogramming AF cells. So those are an interest in addition to FOXF1 in vivo with the NP cells. We're looking at synergistic effects as well. And then we'll also have a major interest in functionalizing the EVs. And this really provides a cell-specific therapy where we can target the EVs to specific cell types from the disc. So now I'm going to transition to talk a little bit about targeting another tissue of interest, so the DRG. So this is really hot off the press, very new. It's unpublished, very new data for us, but this is in collaboration with my EB and nanotechnology expert, Natalia Higuita Castro. So what we're doing now is we're developing EVs with neurogenic cargo. So these are cargo that we hope to target the neurons. And so here's just some data showing that we can package. So ASCL, BRN, MYTI are developmental transcription factors important for neuronal growth. And so what we're doing is developing EVs that can package those factors. And then what you can see here is that when we package these EVs, well, they're preferentially taken up by other neuronal cells. So they show some preferential homing. And then when we treat cells, treat EVs with fibroblasts, basically what we're seeing is these neuronal cargo or neurogenic cargo will actually cause these fibroblasts to take on a pro-neural phenotype. So we're starting to look at like getting these reprogramming effects. And then we have delivered these to the sciatic, via the sciatic nerve to the dorsal root ganglion. And I just want to show you here, here's the DRG. And you can see we get labeling of the, or EVs are labeled in the DRG here. that has been where the sciatic nerve has been injected, and then we quantified the expression of our transcription factors in the isolated DRGs, and we are starting to see, so our ABM factors, which is this cocktail here, we're seeing an increase in these factors in our DRGs that have been injected with the EVs. So really encouraging, kind of a new area from us, and also a model for maybe targeting therapies towards the DRGs. Okay, so now I'm going to transition to talk a bit about another model we're really interested in, which is the preclinical models, the dog models. So as we know, large animal models have a lot of relevance, right? And especially in terms of translation. So typical models that have been used in terms of disc degeneration and back pain have been the goat, sheep, and pig. And in terms of the goat, that's really been pioneered by Lachlan Smith's group and Sarah Goldbrand. And then also Stefan Zeiter has looked at the sheep as a potential model. And then these models are great for examining therapies. So looking at stem cell therapy in the goat, and then also a pig model has been used to explore, human iPSCs, differentiating them into notochordal cells, and then can they survive in a pig degeneration model. So a few years ago, we highlighted a potential companion animal as a model. So this was in collaboration with one of the veterinarians I work with, but the chondrodystrophic dog. So chondrodystrophic dogs are your beagles, your dachshunds, and what's really interesting about them is they can develop back pain spontaneously. And so you can imagine that you have a patient population that also develops back pain, right? So this is of high clinical relevance. There've been studies by Mariana Trifonadu's group kind of looking at the effects of anti-inflammatories on pain. So really showing the utility of this model. And then what's really interesting is a few years ago, they highlighted this mutation, this FGF4 retrogene, as being responsible for chondrodystrophy and intervertebral disc disease in these dogs. So as I said, these dogs, the chondrodystrophic dogs, they develop back pain, disc degeneration spontaneously. You can actually assess pain behaviors. So one of the challenges we have with, say, goats, pigs, and sheep, it's really hard to measure pain in these models. However, the chondrodystrophic dog offers a population where you can assess these both clinically as a pet population and on the research side. And they're really attractive in terms of you actually have a patient population where you can start to conduct preclinical trials. So really highlighting their utility. And then in terms of similarities to the human, people have used modified Thomson grade and Furman grade to assess degeneration in these models, and they have very similar cellular composition and structure and function as we do as humans. So very attractive as well. And then, as I mentioned, when you compare them to other animal models, so here's just a table of different experimental models. We have the dogs here, rodents, rabbits, cattle, pigs, sand rat, goat, and sheep. And then we have the different characteristics of healthy degenerate discs. And what I want to highlight is both the chondrodystrophic dog and another species called the non-chondrodystrophic dog. But what you'll notice is they are the very few out of all the species where they're actually a clinical patient population, right? So very attractive for us to use and high relevance. So a major characteristic of this chondrodystrophic dog is the fact that it demonstrates modic changes. Now, what are modic changes, you're wondering? So typically, these are discs from control, healthy dog. You have high signal intensity, the vertebrae look great, everything looks good. But basically, with degeneration, we have these abnormalities in the subchondral bone, right, which are called modic changes. There's different types, but usually it's associated with inflammation, fatty marrow lesions, sclerosis, so a lot of pathologies. And it's mimicked in human people, so people have these modic changes. And so what myself with my collaborator Sarah Moore wanted to explore is, okay, we have these dogs that are patient population. How similar is it to, like, how similar are these modic changes you see in people? Are they prevalent in the dogs as well? So we looked at 104 pet dogs. We had healthy, normal, we had our chondrodystrophic dogs, but we also had non-chondrodystrophic dogs. And so non-chondrodystrophic dogs differ in that these typically don't develop back pain spontaneously. They develop it with age, and they have a developmental cell called the notochordal cells that they retain. So there's subtle differences. So we looked at the distribution of modic changes within the different groups, looking at modic type 2 and modic type 3. Modic type 2 is more inflammatory, fatty marrow lesions, and type 3 is more sclerosis of the bone. But what I want to highlight is for the control dogs, for type 2 and type 3, we're really seeing very minimal kind of distribution and presence. For the chondrodystrophic dogs, so looking at this level and the level above, you can see it's mostly type 2, a little bit of type 3, but mostly type 2 modic changes. And then for the non-chondrodystrophic dogs, we see that it's some type 2, but mostly type 3. And what's really interesting is that type 2 modic changes are actually the modic changes that are most prevalent in humans. So it's suggesting that the chondrodystrophic dog has modic changes similar to the humans, but in the other dog population, the non-chondrodystrophic dogs, it's mainly type 3. So just further highlighting the similarities with the human condition and the importance of this model. So to characterize this model a little bit more, we wanted to look at kind of the cell level in degenerate tissue, see if it had similarities there. So we looked at the tissue from the chondrodystrophic dogs, including like healthy tissue. And what you can see here, this is Alcium blue, Pica Sirius red. So our autopsy tissue, lots of proteoglycans, looks great. However, from the dogs with back pain, you can see more more collagen, less proteoglycan, and then kind of granulation tissue in the surgical specimen. We quantified the blood vessels within the tissue and expression of CD31, which is an endothelial marker, and we saw increased expression in the dogs with back pain versus the healthy controls. We looked at immune cells and then also saw increases in mast cells and macrophages in the surgical specimens versus the autopsy control. In terms of looking at the interactions of immune cells, so we know that immune cells play an important role in human, and we wanted to see, okay, are we seeing similar things with the dog? We used this co-culture model. So we took just waste tissue from a tissue share where we could isolate healthy NP cells from the dog. We then co-cultured them with canine mast cells, and then looked at the effect of the disc environment. So looked at the effect of different stiffnesses and wanted to see how the cells behaved. So basically this is to be expected, right? So the effect of stiffness on NP cells, so you increase stiffness, you see an increase in inflammatory markers. So we did, so 4% agarose induced an increase in a different array from NGF, IL-6, stem cell factor, and VEGF-A at both 14 and 21 days. With mast cells, we saw a similar trend where you increase the stiffness of the environment, you're getting an increase in all these pro-inflammatory cytokines. And then in terms of co-culture, again, when we looked at the effect of stiffness on these cells together, it seemed to enhance the pro-inflammatory cytokines that were produced. So showing a number of similarities that, you know, you have a stiffer environment, it increases inflammation. So a lot of similarities to the human condition. So this is in vitro work, but we also wanted to look in vivo what was happening. And so this is work led by a graduate student, Mary Hyman. And so in this in vivo study, we wanted to assess and develop parameters to look at structure and function and phenotypic differences, both in the discs and in the neurons, the DRG neurons. We also wanted to assess pain behaviors associated with this model. And also we wanted to overcome some challenges. So a major challenge is that for the dog, there's really not many protocols already developed to characterize neurons and also to characterize pain. So a real emphasis of this study was to explore and develop research tools. So we had female retired breeders. They were about 3 to 4 years of age. And then we induced a puncture to accelerate injury, and we compared it to pre-op or internal control groups. For outcomes, we did MRI, fluoroscopy, histology, and mechanical testing. And then in terms of pain and inflammation, we did activity, we did von Frey, looking at mechanical allodynia, we did electrophysiology. So we isolated the neurons and performed patch clamp to look at sensitivity. We then performed RNA-seq transcriptomics on the DRGs as well. So doing a real deep phenotyping. So I'm just going to show you a subset of the data from this study, but basically we got pre-op MRIs from the cohorts of the 2 cohorts of our dogs, basically showing that the grades were mildly degenerate, which was great. That's what we were targeting, grades 2 to 3. And then in terms of post-injury assessment, we performed T2-weighted MRI, and basically we're able to successfully assess changes in tissue hydration. And with the puncture, the injury, we saw a decrease in tissue hydration. So this was really encouraging. And also something we noticed in a lot of the dogs are these like calcification deposits that were present also in the dog model. We then performed ELISA on the serum, so we could measure creatinine reactive protein as a biomarker in the blood over time, and we could track that. And then also what we were able to do is we were able to isolate and patch the canine neurons to look at changes in hypersensitivity. So we're able to get viable cultures, patch them, and actually get interesting output, reliable data from these. That was pretty exciting. And then last but not least, we performed kind of this global transcriptomics. We optimized protocols to obtain high quality RNA from these dog DRGs, and then we use them for transcriptomics. So here you can see nicely, like as visualized by the heat map, we see major differences, consistent differences across dogs between injured and control groups. And then here's just a volcano plot just highlighting the differentially expressed genes. Interestingly, in terms of upregulation, you can see here on the right, so everything highlighted in red, neuropeptides, neurotransmitters, ion channels, all the things we'd associate to be upregulated in hypersensitive neurons. And then in terms of downregulation in the neurons, we saw these HOX family of genes. And these are interesting. They're associated with development. And so major downregulation of these developmental genes. And so in terms of just doing an IPA, definitely seeing a lot associated with the Krebs signaling, and Krebs signaling basically is associated with injury-induced neuropathic pain. So what we seem to be seeing, at least this data set seems to be highlighting, is kind of a neuropathic pain phenotype, at least in these dogs. So that's kind of the dog model, but clearly there's challenges, and this is a dog model. This is a work in progress for us. We also assessed behavioral outcomes. So we did von Frey, looking at mechanical hyperalgesia. So it's just applying a force to the paw, and you look at paw withdrawal. And then we did general activity as well. So we saw trends in behavior, but it was very variable. And we were kind of disappointed, but we have to acknowledge the factors that are outside our control. So unfortunately, our dogs were housed right next to the primates. And so they were not happy at all. And so this actually affected our behavior because we were testing right next to them and the dogs didn't feel comfortable, right? And also enrichment and handling by the staff, that was out of our control and it would vary every day. And I think these kind of factors impacted how our behavioral data, the outcomes. And so I think moving forward, we probably need more control over these parameters to get reliable data. Another factor with doing large animal research is sample size and cost and accessibility. So it's not cheap to do this study. It costs a lot and obviously it has ethical implications. So there's a lot of factors you need to consider. In terms of improving our pain behaviors, we're looking at things like gait analysis, which we think could give us more reliable data. And then when we're looking at the patient dogs, our goal is really to start looking at client reported outcomes. So as patients fill out surveys, the owners of dogs can fill out surveys to give us an idea of pain as well. So this is kind of summarizing some of the, at least in the dog side, some of the challenges we're currently dealing with, but definitely a very useful model for us. Then, as I described in terms of animal models, there's clear advantages of using the rodent in these preclinical models, but we need to balance this. And a recent publication by the spine research community was reviewing kind of in vivo models versus organ culture. But like I said, it really depends on the question, and there's advantages and disadvantages to using both. Clearly, in vivo models, we have great ways to look at pain evaluation, imaging, and the translation is higher. We can look at tissue crosstalk, we can look at systemic effects, long-term effects. And then on the flip side, organ culture offers another alternative for us to use where many parameters can be assessed in a more controlled way, and you can use human tissue. So just wanted to highlight this and some of the advantages and disadvantages. So how am I doing on time? Okay, still.
Okay.
Okay, good. So I'm just going to finish off. I've got just a few more slides, but now I'm going to transition on some work I know that Michael will be interested in is like the cartilage end plate. So some human models to study the cartilage end plate. So what is the cartilage end plate? So it's this underexplored tissue, right? But it exists here. So we have our lovely disc in the middle. It's sandwiched between the cartilage end plates and the vertebrae. So what does it do? Why is it important? So it basically functions as a mechanical barrier to contain the NP. However, in degeneration, what we see is if you get if that barrier is compromised, the NP will actually herniate out. So this can be a real problem. So in addition to being this mechanical barrier, it's really important for nutrient waste exchange, right? So whether it's the transfer of nutrients from the subchondral bone to the nucleus pulposus, or vice versa, the metabolic waste out to the subchondral bone. So it has this major role. Then in terms of what do we know about end plate cells? So very early on, some early studies back in 1996 highlighted that these cells are active. They're secreting extracellular matrix proteins, collagens, proteoglycans. And then more recently, we've come to understand that they exhibit degenerative effects, right? So with degeneration, we see changes in matrix synthesis. They take on a pro-inflammatory phenotype, and calcification is really important. Calcification-induced degeneration. So clearly there's a lot going on here, but still remain pretty underexplored. And then in terms of some of our work, we've developed some in vitro culture models looking at hypertrophic differentiation in these human cartilage cells, showing inflammation and kind of this catabolic phenotype. We've then done some characterization at the molecular level, looking at the different components of the disc joint and comparing the different tissues. And cartilage end plate cells are very different from articular cartilage. They have a different phenotype. And I think we always assume that they're just very similar to hyaline, but they actually, at the molecular level, pretty different. And then comparing species, we see species differences in the cartilage end plate between, at least from our study, the human the bovine and the canine models. So again, when you're picking models and you want to study the end plate, you really have to think about this. But for us and our purposes, we're fortunate we can access human tissue. So we really focused on the human tissue sources for the cartilage end plate. So in terms of markers, end plate markers always get, the typical markers that we look at for articular cartilage or for NP, right? The kind of classic markers. However, no one's really done a deep dive or deep phenotyping of the actual markers that the end plate cells express, right? So there's a real need for discovery, and to better understand these cells, we really want to understand what markers they express and what's specific to the end plate. I think we've done a nice job as a community looking and identifying NP specific markers, AF specific markers, but there still seems a lot of work to be done with regard to the end plate. So our group performed a transcriptomic analysis of the end plate. So we performed bulk RNA-seq, looking at kind of a broad gene level comparison of end plate cells from healthy degenerate discs. We cultured the cells, looked to isolate the RNA, and then mainly focused on grade 2 and grade 4 cells. We then performed single cell RNA sequencing on the same healthy and degenerate cells. And this was really to look at cellular heterogeneity within the end plate, and using single cell to get the gene profile of each individual cell. So in terms of our bulk RNA sequencing, we identified 53 differentially expressed genes. 38 were associated with the degenerative phenotype and 15 were associated with the non-degenerative phenotype. And then what we wanted to do was actually look and see, okay, out of those genes, how many had been previously studied with respect to the end plate, the disc, and cartilage, articular cartilage in general. And so this Venn diagram just shows basically the number of genes overlap. So we found 2 genes, ADAMTS5 and SORF1, that were expressed in all 3 or had studies that were previously associated with the end plate, the disc, and cartilage. We found 9 genes that were similar between the disc and cartilage, and then 20 genes that had previously been studied in cartilage. But what did this mean? This meant that we actually had 22 novel genes that had not been previously studied in the end plate, disc, or cartilage to probe. So this is further work that our lab is doing, is starting to validate these novel genes. But then in terms of understanding the heterogeneity, so we did bulk RNA-seq to give us that kind of broad level view. Now we wanted to do a deep dive into the cell clusters and the cell populations that existed within the cartilage end plate. So again, we had our healthy or non-degenerative samples. We had our degenerative samples. We identified 9 different clusters in our healthy group. However, in our degenerative group, we isolated or identified 11 clusters with hypertrophic chondrocytes and this other unique multi-stem cell population that was not present in our healthy group. And then when we looked at the cellular composition, you can see that the chondroprogenitors make up the bulk of cells in our healthy and our degenerative group. This is then followed by chondrocyte population 1, chondrocyte population 2, and then 3, and then so forth and so on. But that's just giving you an idea of the distribution. Excuse me. Next, we just wanted to look at, okay, those genes we identified from our bulk RNA-seq, those 53 genes that were significantly differentially expressed, how do they look in our single cell analysis, right? So here we have all the genes that were associated with the healthy, non-degenerative phenotype, and all the genes here that were associated with the degenerate. And we wanted to look at expression across all our different clusters, both degenerate and healthy. But what I want to point you to is this group here. So these are all our progenitor stem cells. And what you'll notice is that all these progenitor stem cells express to a higher level all these healthy genes. However, they express less of the degenerate genes. So interestingly, just like broad takeaway, is that these progenitor cells express healthy genes and they're upregulated in our RNA, bulk RNA-seq, but these are downregulated in our degenerate group. So prior work, what does this mean related to prior work? So MSC-like progenitors have been identified in the end plate. So these are anti-inflammatory, can promote pro-chondrogenic expression by disc cells. So it just highlights a role for these cells and more exploring that we need to do, right, related to the cartilage end plate. The fact that these have large populations of progenitor stem-like cells. So in terms of future work, our lab's probing is exploring some of the novel genes and trying to understand a little bit more about these progenitor cells and what they mean. Okay, so final slide and wrap up. So just to wrap up in terms of mouse models, I want to highlight their advantages and limitations. We use the mouse injury model in terms of looking at safety and efficacy of our non-viral gene delivery systems. It's not perfect, but for the questions we want to answer, it works very well as an in vivo model. Preclinical models have a lot of advantages, especially this chondrodystrophic dog that I highlighted to you today. It's a patient population, and it provides rationale for being able to utilize this model. However, there's still a lot of work that needs to be done in terms of characterizing its pain phenotype and its relation to human back pain. Last but not least, human models offer a lot of utility and promise. We're using this to look at the cartilage end plate. But again, there's a lot of characterization that needs to be done. It's a very important tissue that's been underexplored for so long. And I think if we're going to design therapeutics, we really need to target the end plate and understand what's going on in the end plate and address disc degeneration there also. Okay, I'd like to thank my team of amazing students, my collaborators, funding source, my family, my dog Frank, everyone. It takes a village. And thank you again for the opportunity. It's been wonderful to be able to present to you all today. And I look forward to some interesting questions. Thank you.
Thank you very much, Devina, for that wonderful presentation. And I'm sure your dog would be happy as well. Yeah, really wonderful. So any questions?
Absolutely. We have lots of questions.
I thought you might.
Yeah, no, that's fantastic work, Devina. It's very amazing. I really like everything, but especially you delved in quite deeply in the end plate story. So that's pretty cool. If I would go, so starting from the end, then I'll go back to the beginning in terms of questions. So the end plate stuff. So I noticed, like in some of your genes there, you didn't find anything related to inflammation. Like I didn't see IL-1, IL-6, or any of these inflammatory.
Yes.
Yeah, it's that's interesting. I saw ADAMTS5, so I didn't see MMPs, but I saw some ACANases, I guess there. So what's your take on that? Like how?
So it's interesting because we were surprised, and it did highlight, like in terms of the differential expression. So out of doing bulk RNA-seq, we identified 53, which we were surprised at how low the number was. But then I don't know, given the work that you've been doing as well on using human tissue, right? So we have the innate human variability. We had a sample size of 4 for healthy and degenerate. But I think in terms of defining what a degenerate disc, like a degenerate end plate is versus a healthy end plate, we've got no good scoring system. So we can base it on like a healthy disc, but that doesn't mean to say that it's like like the end plate is completely healthy or vice versa. So I think some kind of human variability probably played in a little bit. But I'm wondering if it's more, while we can induce inflammation in these end plate cells, are there additional roles of the end plate cells that we've ignored? And maybe this points towards some interesting, maybe more ECM related roles, or, I don't know if you noticed some ion channels also kind of, so maybe mechanotransduction, maybe ECM. We definitely saw kind of like biological functions like protein translation, mitochondrial dystrophin function. So other, yeah, it wasn't the classic usual suspects for sure.
Mm-hmm. You had the EEA1, which is interesting. That's involved in calcification, I think.
Right. Yeah. So
Yeah,
definitely
so you
some,
have that.
yeah.
Yeah. Yeah, I think so. That was
We can
pretty
talk
cool.
more as
Yeah,
well, right?
yeah. The other question would be post-translational modifications. Obviously, with these techniques are amazing, but they also don't address like what happens to proteins. So that like you could even extend further your study in like a
Yes.
proteomics. So
Yeah,
it's like this.
Yeah, that's what
Uh,
so we have an interest. I didn't present it today. It's some of the current work we're doing is, we have an interest in extracellular vesicles as a delivery system, but also we're trying to characterize the secretion from the end plate cells. And see what the role that these vesicles play in paracrine signaling within the disc joint. And so we're actually my student Carl's doing proteomics on the EVs that are derived from the end plate cells to get an idea of what they're secreting specifically. So I think that'll shed some light on some, at least the protein level changes for sure.
for sure, yeah, that sounds very interesting too. Looking forward to that. And so I'll just go back to the nice work, very nice work on characterizing the chondrodystrophic, non-chondrodystrophic dog model. That was quite impressive. So it gives insight onto how you're going to use for disc degeneration. What, more details is better when you're trying to characterize and, to understand the findings when you're doing treatment, which is pretty cool. That's just a comment. But if I go back to the mouse, so you had a very interesting, amazing results with your phospho-F1 EV injection in your puncture model, right? The injury model in the mouse.
Right.
So I noticed, and I'm sure you noticed, because that's the nature of your model. After, I think was it a 12-week? You waited 12 weeks before you? Yeah. So the puncture disc showed a lot of fibrosis, right? So it looked like a lot of collagen expression and so on, and of course less proteoglycan or aggrecan. So I was wondering, you're treating, you're injecting obviously at the same time you're putting in phospho-F1 as you're doing the injury. So if the disc had a chance to degenerate, become fibrotic, let's say in your injury model, have you tested, or you plan on testing, or do you think like an injection later on, let's say mid or late stage of degeneration, would that reverse what you see?
Yeah, such a great question. That's actually what we're tackling or have been for the past few months. So as a proof of concept and feasibility, so working with mice is challenging. They're so small, right? The lumbar discs are tiny. And so to optimize the actual surgery, it literally took our grad student maybe 5 or 6 months. It took Nina to optimize just the surgery to do it, right? And so in terms of feasibility, we thought, okay, if we can inject and injure at the same time, at least as a proof of concept, that will give us an idea if our intervention will have some sort of regenerative effect, right? Right now we can see that. Our next goal is to look at a more relevant, like its relevance to the clinical situation, right? Because clearly, we don't have an intervention at the time it's injured. That's not possible, right? So to your point, can we intervene in after 12 weeks, right? So we see this fibrotic response. And so we're trying to optimize the second surgery, and it's not trivial on the mouse, to be honest. So it's quite challenging to come in with a second surgery. You've got scar tissue and the mice survive, but it's hard. So what we're trying to do is think of alternatives, right? What are there other models? And so this has led us to probe maybe kind of an age-related model, or maybe if just even the spark null potentially, right? Look at our intervention on something where we've got some kind of degeneration that is not associated with an injury, where we can intervene. Because I think the mouse, it's a really great, but it's challenging to go in with the second surgery. And to your point, that's exactly what we're dealing with right now.
Well, thank you. Yeah, very good though. Very impressive. Congratulations.
Well, thank you. Like I said, it takes a village. Like you need really hardworking graduate students. I'm sure there's many of you here, right? And it's all work from you all that contributes to what I'm presenting. I am fortunate I get to present. It's really the students that do the hard work. So really grateful to have like passionate and motivated students that just are excited and like to nerd out like over science like me. So it's awesome.
Yeah, very nice. Thank you.
Okay, I guess I can see a question. Is it Marshid? Is that right? How do you perceive the effect of sex of animal models in the induction of the injury? That's a great question. So specifically in the induction of injury, we are seeing different effects, right, based on whether they're males and females. And literature has shown that pain phenotypes are different between males and females. In terms of the underlying mechanism, I'm not completely sure. We haven't really probed that. We've just looked at the different groups individually. But it's a great question. And I don't think there's enough studies that have been done to characterize the like males and females separately in discogenic back pain research. And I think hopefully more's to come where people's research will focus on that. But great question. So thanks for that.
So Devina, that was really amazing. I really like your research. It's really wonderful, and the directionality is really great way. So I was intrigued by the work with the vesicles, and when you are looking, especially the new ones, and the sex differences shows that the results that we get, we have to be cognizant of that.
Right.
Yes, and that's one thing that you are showing. And perhaps it's quite tough to extrapolate when you go to dogs because it's not as easy as to get, female, male.
Right. Yeah, it becomes harder. I mean, we're hoping to do it where we can look at males and females. I just presented work from female dogs because those were the ones we're able to have access to. But yeah, I mean, the goal would be to be able to look at both sexes. I think with any study, being able to look at the sex effects is important, and age as well. I think not just sex, but looking at age. I mean, we're using mice that are 15 weeks. If we used mice that were maybe a year old, would we see the same regenerative effects? I'm not sure. So that's also something that we want to look at.
Right. Which leads to a philosophical question that when we see results, we have to see them in that context.
Right.
Because it goes back to the question that Mike was asking for the end plate. Because I was looking for the usual suspects, the collagen types, type 2 collagen, type 9 collagen,
right.
type 11 collagen. I was looking for TGF beta.
Right.
Yeah.
And so I think those were we didn't separate out by sex, we didn't separate out by age. So I think it would have been nice if we could do that, but again, with human tissue we are limited as well, right? So but I'm wondering if that is feeding into some of the variability that we did see as well.
Right. Because I'm sure if you had a lot of different samples from male and female, different age groups, as you said, yeah, and we could be able to see quite a picture. Because the fact is, the type 2 collagen should be there.
Yes.
Type 9 collagen should be there.
Yeah.
And type 11. So those.
Right. And so, yeah, if we go back to our just analysis, the collagens were expressed. It's just the differential expression between healthy and degenerate that we didn't see. And those were just the genes I highlighted. Those are the only ones that were differentially expressed. And I think, as well with the age versus the sex, I think we don't have a great grading scheme for what a healthy versus a degenerate end plate is. We'll just use usually the Thompson grade or Furman to be like, okay, this is a healthy disc, but it doesn't mean it's a healthy or, the disc is, the end plate is completely healthy, right? So I think, that's something that as a community, it'd be great to get other people's feedback on, like, can we get a really good grading scheme for the end plate?
Right. So your work looking at the DRG, I think Anne Marfet, she showed that when you have the painful signals, that they are generated in the peripheral tissues, and then these are modulated in the DRG.
Yeah.
And then from the DRG, this is carried to the ipsilateral dorsal horn.
Right. To the,
and where you get this neuronal microglia that then contribute to pain. So your intervention at the DRG level would be interesting when you have a signal coming.
Yes.
And then go to EV and then see if that signal is transmitted to the central nervous system.
Yes.
Yeah.
Yeah, for sure. And so that's some of the rationale. We're trying to see, and it's tricky with pain, right? We don't know, like, especially with chronic pain, whatever we do to the disc, is it going to be enough? And so that was the rationale for starting to look at the DRGs and see if we can develop any drug delivery systems for the DRG, whether that would be more efficacious in terms of targeting some of those pain signals, rather than just relying on something in the disc to do everything.
Right. Yeah. You could look at the chemokines, what she's MCP1 and CCR.
Right.
Yes.
Yeah.
I think in that wonderful paper in Proceedings that she showed, there's you need MCP1, CCR2 for the maintenance of pain throughout.
Pain. Yeah.
That would be nice to see if that is shut down.
Right. Yeah, for sure. Yeah, that would be great. That's a really interesting target that we could potentially look at. Yeah.
Yeah.
Yeah, that'd be really cool.
Any questions, anybody?
Yes, Dr. Faxon, fast question. Thank you, Dr. Devina, for this very interesting lecture. Actually, at the end, you said that we have 22 new genes not previously published. So we have the information, but you mean the other one which we have it between CEP and IVD cartilage, we have publications.
Right, that's correct.
But no one studied this.
Right, so these two that haven't been identified in previous literature related to the end plate, disc, or cartilage. So those we're hoping to do a further validation just in more end plate samples to kind of look at their expression. Because one of the things I didn't highlight is that there's no great novel marker, or no one's identified a novel marker for end plate cells, right? So we have some unique markers for nucleus pulposus, we have some unique markers for annulus fibrosis, and it's been great, right? We've been able to create genetic knockouts that target NP cells, that target AF cells, but how are we going to target the end plate? And so, one of the reasons why we decided to pursue doing some transcriptomic analysis was really to try and identify, look at and see if we can identify novel markers that we could use, in terms of trying to understand the end plate a bit more, because I think the end plate often just gets associated with articular cartilage and it's just very different. So.
Thank you.
So we have a last question from in the chat. Muskan?
Okay.
Yes, I'll just say. So, great presentation. I really loved it. And yeah, and there are many different spontaneous models you discussed, but I'm more into the co-culturing. You said
Okay.
that,
So, yeah.
yeah, I was like, you said like you co-cultured the disc cells with the macrophages and saw the microenvironment, and saw the degenerative changes. So was there any polarization of macrophages into M1 or M2 or something? Because in the inflammatory, condition?
Right. Yeah. So for this study, for the data I showed, it was mast cells, so it wasn't macrophages. We have
Oh.
looked for macrophages in the dog, but just the research I presented was on culturing of mast cells.
Okay. Thank you so much.
Yeah, sure.
Oh, I thought it was macrophages, so
Yeah, no problem, no problem. Very similar, but yeah, mast cells, they release granules that contain a lot of inflammatory molecules. So
thank you so much.
yeah, thank you.
Sorry, Devina, one last comment actually just
Yeah.
popped my head. I was just wondering when you're doing the end plate, when you're isolating for, let's say, your RNA-seq and so on, bulk RNA analysis on the end plate. So you have a mixture you found, which was interesting, a nice different populations of chondrocytes. Are there, because we know that end plates are infiltrated with dendrites of neurons. And these dendrites probably contain some RNA, RNA molecules within. So when you're doing your isolation, is it possible that when you're also finding these, upregulation of markers associated literally with neurons that are present within the tissue?
Yeah, so it didn't come up. We saw like osteo related markers, but nothing neuronal. That just didn't come up at all. So that was surprising, because if we're thinking of innovation, right, it's either going to come from the annulus or the end plate, especially in degeneration. But we didn't see anything related.
You saw something interesting though. If you go back, I think you saw SSTR1. If I'm not mistaken, that's a somatostatin receptor,
right.
the receptor for somatostatin, the neuronal peptide. I worked a lot on this, so yeah, that kind of popped in my mind when
we should talk more offline.
Yeah,
right. And I'd be happy, like the paper's out. So if you want to chat a bit more about potential markers, I'd love that. That would be
sure, yeah.
It'd be really cool.
Sounds great. Thank you. Yeah.
Yeah. I think if we're not gonna end.
We just keep talking and talking, right?
Yeah, so it would be nice to see, there's work that's been shown that of annexin 2 in the end plate, and that is it'd be nice to see how the annexin 2 that it's related to calcification in the end plate.
Okay.
Yeah, it'd be nice to see in your wonderful to see how annexin 2 features.
Okay.
Yeah, that would be nice.
Annexin 2. Right.
Yeah. So yeah, thank you very much, Devina, for that really wonderful work, and Kadi, nice work. Continue. You are in the nice lab, the best lab in the world.
Thank you so much. Yeah, thanks so much for the opportunity. It's been really fun.
Yeah.
Nice seeing you. Yes.
Yeah, you too. Hopefully see you in the flesh soon.
Yes.
And thank you very much, everyone.
Thanks, everyone.
Yeah. Bye.
Bye.